So we just finished learning about the integumentary system, which part of which is the skin, and part of that skin involves tissue that is no longer alive, right? Your outer layer of your epidermis is just flattened cells that really aren't receiving any nutrients. A similar question was posed to me as I was defending my master's degree, and one of my committee members asked me if bones were alive. Well, and to answer that question, you just have to kind of know the structure of bones. Yes, bones are hard and they resemble rocks and things of which we think are not alive, but they still hold the characteristics of living tissue. They have a blood supply, they have cells which rely on that blood. They just also have a very rigid framework in the bone tissue, which is necessary for its function, because our skeleton, our bones, act as a scaffold or a support system for the rest of our body. So the main functions then of our skeletal system are first for support, movement, and protection. Of course, it's not independently able to do that. There is also a place for mineral and storage of energy. So you have fat, which you can see in the yellow bone marrow. Red bone marrow is where blood cells are made in a process called hematopoiesis. And you have calcium, phosphorus, zinc, other minerals which are in bone, which can be mobilized if they are low in circulation. And finally, there are some hormones which are produced in the bones, including a bone called osteocalcin. There are a few different types of cells that you can find within bone, active bone, living bone tissue. First is our stem cell, our osteogenic cell, which is found in the periosteum and endosteum. This is the area of the bone in which growth occurs, and these new bone cells are made. They are mitotic, meaning they can divide, and they can also mature. One of the cells they mature into is the osteoblast. The osteoblast is also mitotic, meaning that it can divide and make more of itself. The osteoblast also secretes the unmineralized bone matrix collagen, so it does not have the calcium and phosphorus part of it. And collagen by itself does not form, it gives the flexibility of bone, but does not form the hardness which is needed through the salts. This is called osteoid, the matrix. When they become trapped by matrix, the osteoblasts, they then mature into osteocytes, which are our third cell type of bone. These are non-mitotic, meaning you can't make more osteocytes from the current osteocytes. They mature from osteoblasts. And they are surrounded by bone matrix and then matured. They do have a function still. They communicate with osteoblasts and osteoclasts and seem to coordinate then those cellular functions. Osteoclasts, now these are not, they have not derived from the stem cells in the bone. They are instead derived from white blood cells called monocytes. And that is fitting because their function is to absorb bone material and essentially eat at it to break it down, or release minerals, or reshape bone. So for example, pregnant women lose bone density, generally if they don't have enough calcium in their diet, and that is to release calcium into the bloodstream. Osteoclasts are responsible for that. That calcium then can be used for the development of their fetus. These can all be found in a similar area within bone tissue then, our osteogenic or osteoprogenitor cells are going to be floating in the endosteum or periosteum areas where new bone tissue or new bone cells are going to be needed. Osteoblasts then are going to be secreting the bone matrix, the osteoid or the collagen, and then as they fully surround themselves in that, become mature osteocytes, and then you have osteoclasts. You can see these black parts then are the nuclei, which are going to eat away at bone tissue where needed. All right, bone is made up of, like we said, the matrix is made of collagen. 35% of it is collagen. And that gives bones flexibility and tensile strength, or the ability to withstand bending or stretching. And bones are very strong without being brittle, but it doesn't have that combination unless they have the calcium and phosphorus and other minerals embedded within it. Bone is half as strong as steel against compression and equally strong as steel in resistance, so being able to be pulled apart. Older people generally stop producing collagen at the same rate as younger people, and so their bones will become brittle. They may also have problems with salts and calcium and phosphorus absorption. And that then can produce lighter and weaker bones with more space in between the bone tissue. A normal bone will have a certain amount of density of bone tissue and space in between it. If you aren't getting enough fluoride, calcium, or vitamin D and fluoride are important for the regulation of calcium and the laying down of the bony tissue. Smoking can also increase the lack of absorption of these vital nutrients, and decreasing exercise can also prevent a rigid bone structure. All of these things can then lead to osteoporosis, which means porous bone that has too much space in between it. Some medications can be used to increase bone mass, as well as changing your diet and increasing exercise. All right, there are then fundamental units of bone tissue. And the fundamental unit for compact bone is the osteon. Now, an osteon is one of these circular rings and everything inside of it. It contains the lamella, which is the bone rings. It contains the lacuna, which are the little spaces within the rings where an osteocyte will be housed. There is one big central canal at the middle, or Haversion canal, which is going to have these vertical running blood vessels. And then you have Volkmann's canals, which are going to be connecting blood vessels horizontally or perpendicular to the central canal. You also have little nutrient canals for nutrients, and you can't see this, but that's O2, so oxygen, called canaliculi, so little canals essentially. And those are going to be then connecting then the rings or the osteocytes between the rings. All right, so that is an osteon. You get these together and they form bone tissue. Trabeculae is a different type of bone. It is literally means little beams, and this is much more porous bone called spongy bone. And all bones have areas that have both compact and spongy bone, but the composition of each or the proportion of each depends on the bone morphology. So where it's positioned in the body and what is its shape and function. So some of the bone morphologies that we have 5 different kinds. Long bones, these are your kind of typical bones that you think of, such as the femur and the humerus. They are long. Short bones, such as the carpals, these are bones within the hands or feet. Flat bones, which are like the bones of the skull. Irregular bones, such as the vertebrae, they have kind of all sorts of different shapes within them, and sesamoid bones, such as the patella. Flat bones have a thin layer of spongy bone called dipole, which is then sandwiched between two thin compact bone layers. And some examples include the skull, sternum, scapula, and ribs. Irregular bones just have odd shapes. There's no specific shape for them. Examples include vertebra and the hip bones. And this is also a thin layer of spongy bone covered by compact bone. So similar to the flat bones, but just in different shapes. Short bones have a thin layer of spongy bone as well, covered by compact bone. But they are more cube-shaped rather than flat or irregular shaped, and include the bones of the wrists and the ankles, the carpals, and the tarsals. Long bones have areas on the end, so these are generally the moving parts of your bones, of your appendages. And they have an articular cartilage point, which usually is at the joint up here. They have an epiphysis, which is the end of the long bone, which is mostly made up of spongy bone and has bone marrow, generally red bone marrow, in there. Long bones also have a shaft called the diaphysis, so the long part of the bone, not the end, is mainly made of compact bone, and then has a cavity called the medullary cavity, in which is the yellow bone marrow, which is mostly fat. Okay, fat, which is essentially storage for energy. There is also a couple of structures which we already mentioned, including the periosteum, which has two layers: an outer layer, which is mostly dense connective tissue, irregular connective tissue, and an inner layer, which is osteogenic, where you have those osteoprogenitor cells or osteogenic cells, which become osteoblasts, which become mature bone cells. There is also a rich supply of nerve fibers and blood vessels that need to be in that area to provide nutrients for these newly developed cells and stem cells. And the periosteum, also the fibrous layer, allows for the attachment of tendons and ligaments, which are types of dense connective tissue, which attaches to muscles. The endosteum then is just inside of that, also has mostly osteoprogenitor or bone cells, which are stem cells, right? Osteogenic cells. And that lies the inside of the medullary cavity. All right, so those are the different sections of the different types of bones. But bones, don't start out in your body, of course. They are formed over time, and there are two different patterns of ossification. The first that we're going to go over is intramembranous ossification. So intra means within, and membranous is referring to a membrane. So it's occurring between a membrane. The first step is the mesenchyme, which we all know is the precursor tissue to bone cell, differentiates into our osteoblasts. And the osteoblasts then can secrete osteoid at what's called the ossification center. Okay, these can occur at the same time in different layers of a mesenchyme. The osteoblasts then secrete this osteoid until it creates a mature osteocyte, and that osteocyte then is in a calcified area of bone. This bone then reshapes it into what's called woven bone, and it is laid around and within different blood vessels until trabeculae, which was that fundamental unit of spongy bone, forms, and then the periosteum is formed on the outside. And finally, you have compact bone replacing woven bone on the outside and inside, and this vascular tissue on the inside becomes red bone marrow. So these are the microscopic steps and macroscopic steps that you can see. So microscopic steps, of course, you can't. These are happening at the cellular level, and then the visual ones you can see the macroscopic level. This is the figure from the book where you can see these. You first have the mesenchyme and end sheets of mesenchyme ossification centers occur as these osteoblasts secrete osteoid, which eventually becomes little bony tissue, which then these cells develop into our osteocytes. And as these ossification centers then develop more and more, they then create this trabeculae and woven bone where blood vessels go in between the spaces, and the periosteum then forms on the superficial or outside parts, and finally a mature spongy bone cavity filled with red bone marrow is surrounded by our compact bone. So intramembranous ossification occurs in flat bones and irregular bones. Endochondrial ossification then occurs in long bones. So the first part of endochondrial ossification is that you have the somewhat resemblance of a bone shape in the form of cartilage, and then the perichondrium forms a collar around what will become the medullary cavity or the shaft. And they form this around. This is a hyaline cartilage precursor bone. The chondroblasts, which are what's producing the hyaline cartilage, in the center then start to die. And then where they die, they essentially kind of crumble away, and it forms a cavity which the periosteal bud forms. So this is where blood supply then is innervated that area, and red bone marrow and bone cells then start to proliferate and create bone. So this is the primary ossification center. So this is where bone first starts to be made. The next step is where secondary ossification centers occur on the epiphyses. So in a similar manner of the primary ossification center. The osteoclasts then start to form the medullary cavity, and finally the epiphyses ossify as spongy bone, and the epiphyseal line forms. So this is a cartilaginous area between the epiphysis and the diaphysis in the long bone, and that allows the bone to continue to grow until it matures. So in a developing fetus or baby, if you will, you have flat bones wherein the intramembranous ossification process is occurring. You have long bones, you can see here, where there's still cartilage in between these bones. These bones have not fully formed yet. So here you have the shaft of the bone, which has its ossification center, mostly fully formed. The secondary ossification centers have not formed yet in these. So you can see different patterns of ossification occurring throughout this developing fetus. All right, so just as you can just kind of see from this chart, our intramembranous ossification patterns occur starting with mesenchyme, and we have our flat irregular bones being formed through that. And in endochondrial ossification, we start with hyaline cartilage, which is how we form long, short sesamoid bones. Some bones only have one ossification center. One secondary ossification center, and some have two. So a really long one like a femur is going to have one here, and then one in the middle. So 3 total and 2 secondary ossification centers. Short bones don't have epiphyses, so they just have the one primary ossification center. At the epiphyseal line, where growth occurs in length, you have a number of areas of cartilage where different things are occurring. In the reserve zone, this is the epiphyseal line, and this is where the chondrocytes or chondroblasts are going to accumulate and allow for the division of more. So this is the mitotic part. In the proliferation zone, this is where mitosis is going to push cartilage cells down, and then you have hypertrophy zone where they're going to actually change shape and enlarge. And then you have two here at the end. The zone of calcified matrix is where the osteoprogenitor, osteoblast and osteoclast cells are going to turn that cartilage into bone, and finally the new bone is going to be laid down in the ossification zone. Okay, so this is what it looks like from the figure in your book with our different zones and what's going and occurring in each area. All right. So these processes are not only important for bone growth and bone maturation, but also your bones don't remain the same shape from when you are a baby to you are an adult. In fact, they change in proportion and shape and everything else. And so bone remodeling requires the continual use of our osteoblasts and osteoclasts and formation of osteocytes. So you can see, like in the end of this bone, it's not going to just push the same shape forward, that actual bone is going to have to be reshaped, and osteoclasts are going to have to do that. Stress also allows for bone remodeling. So if an athlete is using their right arm more than their left, the points of stress through the athletic motion is also going to cause bone remodeling. All right, there are a lot of different structures on bones which also are the result of bone remodeling, and they are for many different things. You have attachments of different ligaments, or maybe joints or connections between different bones. You have cartilaginous areas. You have attachment of blood vessels or nerves, which have to go in and out of different areas. And so all these different facets and tubercles and these things with funny names are important for the function of the bone. And here are a few examples of, found in the femur. This is the part of your elbow that fits into your humerus, and you can see there's just lots of different shapes and parts of the bone, which so for example, the calcaneus bone here attaches to a big tendon which stretches up and down your leg, called your Achilles tendon. Okay, all of these have all of these names are used, you'll find in lab, in a lot of different bones. Some different bones will still have fossas with different names, and this is the description of each and some examples. So this will be important for learning some of the bones in lab. Now, one other area where remodeling is very important is healing a bone after a break. And these are the steps that occur. So after a bone breaks, either partially or full break, you're going to have a rush of blood to that area, and that's going to form what's called hematoma, essentially a large goose egg or swelling area. And that's going to bring a lot of blood to the area, a lot of nutrients and cells, which are important for then starting the healing process. And the first thing that's going to happen is your spongy bone is going to start forming essentially little ossification centers. Blood vessels are going to bring the nutrients to those to allow for growth to continue. You'll then create a bony callus of spongy bone, which then will start to develop the necessary compact bone, or whatever material was originally there. And you're going to have then a an area which is slightly different. It's not going to return to its perfect shape. You may have a over-calcified area, which may diminish over time, but generally, if you have a break, you're always going to have some sort of extra bone in that area. And osteoblasts, osteoclasts, and of course, red blood cells and blood cells and immune cells are going to be important for our beginning steps, and our osteoblasts, osteoclasts, and osteocytes are going to be important for the other steps. So a good exercise is to think about, all right, what cells are important for each of these different steps. All right, so a question for you is, well, if when you break a bone and the space created by that gap fills in with new bone, what if you purposefully broke it and then just pulled it further apart? Could you make a longer bone? If anyone has seen the movie Gattaca, they actually do this because they're trying to impersonate one person as another, but the height is not right, so they break his legs and pull it apart and make him taller, right? So the question is this science fiction, or is this something that can really be done? Well, in fact, it can be done, and it is done in a medical procedure called distraction osteogenesis, which has these 3 steps. The first one is to fracture or break the bone, then slightly move them apart with usually braces to holding them in place, and then new bone will just form between where that gap is made. And as long as you keep it stable, it should form, a longer bone area. Very useful for orthopedic procedures, including height lengthening. So, for example, if you have one leg that's slightly bigger than the other, longer than the other, you may need to go in and correct it to allow for better posture or back problems or whatever. And it's also used for correcting jaw deformities. So if a jaw is broken or maybe just didn't form right, you can go in and break it and reshape it with braces as needed. It looks like a very painful process. I've got a nice link here that shows a little video about the process. Maybe we'll watch that in class. But that's it for the introduction to our skeletal system.